Welcome to the Goat Genetic Improvement webinar series. So this series is going to take place over the next 2 weeks with presentations starting today and also including July 22nd, the 27th, and the 29th. So Amanda Amaratunga is DVM, is the National Veterinary Program Specialist for Scrapie. She's been with Canadian Food Inspection Agency since 2009 in the Policy and Programs branch. And she's been working primarily with transmissible spongiform encephalitis, or TSE. She's worked with chronic wasting disease as well for a number of years, and most recently has taken on the responsibility for the Scrapie file. So outside of work, she loves to spend time outside with her 2 little boys, and as a fitness habits coach for overloaded women. So Amanda, we're happy to have you here today. I will hand it over to you to tell us a little bit more about Scrapies and goat genetics in Canada.
Great. First of all, thank you to Erin and Marlene for inviting me and giving me an opportunity to share a little bit about our national Scrapie eradication program and some of the current and future work that we're doing. We really appreciate the opportunity to have a chance to interface and get to do some outreach like this, especially in this current environment. So I really appreciate that. I thought I'd just do a really quick summary of what I'm hoping to cover today. I do have quite a few slides here. I know Erin and Marlene plan on sharing them. I will go through some of them fairly quickly, but I just wanted you to be able to have them. And I'm going to focus on the key points and also try to leave a little bit of time for questions at the end. What I hope to accomplish today is to help bring everyone to some sort of same level of awareness of what Scrapie is, how it causes a disease in individual animals and farms, how it's spread, what to look for in your herd, why controlling Scrapie is important, a little bit of a summary of our history of goat Scrapie in Canada so far, and then I will shift a little bit more into talking about our genetics and how they tie into the remaining work we need to do and are currently doing to help eradicate Scrapie from Canada and goats. And I'm going to wrap up with some of the current and future work CFIA is doing involving Scrapie eradication in goats and genetics, and also what you can all do as an industry, including producers, to help eradicate Scrapie because of some of the inherent challenges with this disease. We all need to work together to achieve our goal of Scrapie eradication. So with that, I will go ahead and get started. So Scrapie is part of a family of diseases called the transmissible spongiform encephalopathies. And what these diseases have in common, so you can see they impact different variants of the TSEs, impact different species, including humans. We have the CJD or the variant CJD. So these are diseases that affect primarily what we see the most is the central nervous system. And the main characteristics are that they have a very long incubation period. The end point of this, that's not compatible with life, is that we end up with spongy brain degeneration, and the disease is always fatal, 100% of the time. So just a little bit about the agent. It's caused by a rogue protein, the rogue version of the prion protein, which accumulates in many tissues, as I'll talk about. Some of the key ones, though, are lymphoreticular system, which involves our immune system and the lymph fluid that goes through our body, as well as our central nervous system, which is pretty important, right, as far as being compatible with life. And so what happens is that we have the normal prion protein, something happens and it converts into this rogue or abnormal prion protein. The major challenges with this disease that involve really all of us is that it has a prolonged incubation period. So we can have infected farms and infected animals for a number of years and not know it, which is why testing is so important. We have this challenge of environmental persistence. The Scrapie prion can live in the environment, the various aspects of the environment on the farm for a number of years. And the lack of immune response. So unlike bacteria or a viral disease, we can't detect it easily in live animals because of this lack of immune response. That makes it not only difficult to detect, but also we can't treat it. This slide is just here to show you that based on what we know, the Scrapie prion is widespread in almost every major tissue system in the body. And that makes it challenging as far as transmission and contaminating the environment, and also direct and indirect transmission between animals. Having been a TSC expert at CFIA for a number of years, I spent the bulk of my time of my career here with chronic wasting disease. And so in the gamut of TSCs that for which we have programs for in Canada through the Government of Canada, we have BSC on one end that's limited to very select tissues, so it's not as infective. We have Scrapie, traditionally has been in the middle, and then we have CWD, or chronic wasting disease, on the end as far as being the most infective and spreading as easily. What we've recently found out in recent years is we have more widespread tissue distribution, and Scrapie transmits more easily than we had thought. So it's actually closer on the spectrum now to CWD than BSC than we had previously thought. So as far as transmission, how is this transmitted to the individual animal or in between animals? Main method of infection is through ingestion. So they're taking it in primarily orally. And the two key avenues are around the time of lambing and kidding. So prions are excreted in the placenta and vaginal discharges. So any animal that has access to a lambing or kidding environment can ingest the prions and become infected. Neonates are particularly susceptible, as they are to many things. And then the other piece that has come more to the forefront with Scrapie in recent years is that prions, we know because they impact more tissues than we had previously been aware of. They are secreted or excreted, contaminating the premises. So that means when I say secreted or excreted, that means any fluid that exits the body. So the really common ones are milk, feces, and urine. So we can have things like contaminated pastures, barns. Obviously there's the lambing and kidding areas, and the challenge we have with our current science is that we don't have the ability to detect Scrapie in the environment, nor do we have the ability to treat a contaminated environment. I mean, we do as far as cleaning and disinfection, but there's no 100% solution because, of course, farms are large, expansive areas. And so we have had reports of very long periods of environmental persistence. So that can be a big challenge, especially if we may have an infected farm or infected animals and we don't know it. I'm not going to go through all of these clinical signs, but the thing I will bring your attention to that can impact you as a producer is that the disease can have an incubation period of up to 2 to 5 years. And so having this prolonged incubation period is a challenge because we can have animals that don't present clinically. They can be harboring the disease. They can even be shedding it in the environment. They can be transmitting it to other animals and we don't know. The clinical course, so when we see signs in the animal clinically, is fairly short, 1 to 6 months. So by that point, we could have significant spread of disease already. As far as overall herd mortality for the Scrapie flock certification program, we consider a 2 to 3% to be average. One of the things we can look for as far as clues, because we don't have a lot of options as far as Scrapie testing in live animal to see if an individual animal is positive for Scrapie, is to look at herd mortality. So in our past experience with positive herds, we've seen a slightly increased herd mortality, anywhere from 3 to 5%, all the way up to 20% in highly infected herds. And of course, this can be a very costly disease to have. So what can this look like? We see all these lists of clinical signs, and the challenge, because it has a long incubation period and can affect so many tissues, is that there isn't always a very, very specific set of signs, and we can say that animal has Scrapie for sure. And so what we often see, in Europe, there have been reports of pruritus, or sort of that classical itchiness that's associated with the name Scrapie. But in North America, what we've seen more often, what we've learned, is we see just sort of more poor doers. So poor doing animals, wasting. And as many of you know, that's not specific to Scrapie. And neurological and encephalic signs are common. Again, we can't see neurological signs, but it can be a wide range of how it can present. So basically, Scrapie should be considered as a possible diagnosis in any animal with any neurological signs or any sort of poor doer or wasting condition. So I wanted to bring that to your attention because, when you look in the textbooks or you see, information out there, you might think there's sort of this very clear picture. And I just wanted to highlight that there often isn't. It's often very subtle. And that's where testing animals that die, mature animals that die or are slaughtered, is a very, very important tool in our arsenal for detecting Scrapie. Just a little bit of history. It's been around. It's quite an old disease, has been reported, first described in the UK, 1732. First reported case in Canada was 1938. It became reportable in 1945. We officially developed a Scrapie eradication program in 2005. And our goal is to be Scrapie free. So much like Erin was talking about breeding for genetic resistance over time, our goal to be Scrapie free is a goal that we achieve over a number of years over time. And what we want to do is just keep moving in the right direction. Importance of Scrapie control. So not every country in the world is recognized to have Scrapie. And so what that means, because we don't recognize that everybody has it, is that it is a barrier to trade. And so for us specifically here in Canada, it is a current barrier to export with certain trading partners. We have our common trading partners in the US and European Union that are engaged in eradication. And by volume, they have more numbers and more samples over time. They're also ahead of us in their path to eradication. Animal welfare, as you can imagine, this is a really tragic disease if you have it in your herd. And so animal welfare is a very important reason to control Scrapie. Environmental accumulation, as I mentioned, because we have this problem with environmental persistence, we want to try to find all of the Scrapie positive farms and clean this out as much as we can to help prevent future problems. And then, Scrapie is not currently known as a human health risk, but the challenge with these transmissible spongiform encephalopathies is we can get things like strain mutation and there's just also a lot of things we don't know. Humans live a long time. And so we have a lot of long-term research and information that's going on. So while it's not known to be a human health risk, that is always in the back of our mind because it is part of a family of diseases in which humans have been impacted. So just a quick summary of some of the cases of goat Scrapie in Canada. Our biggest challenge, I just want to highlight in this slide and the next, is that we have very little surveillance in mature goats. A couple of challenges with that. is that goats do not officially have identification yet. We do have voluntary ID, which I'll talk a little bit later, and it's in the books in our legislation to go through, I believe, next year. It's in the books. But without identification and enough surveillance in mature goats, which we currently do not have, it's very difficult to get a clear idea of the prevalence of goat scrapie in Canada. And until we have that information and we are able to find all the remaining cases and stamp them out, we will not be able to declare ourselves free of scrapie. And so I just want to draw attention to two sort of niduses of infection in recent years. There's 2013 and 2015. First diagnosis of an infected goat herd, official diagnosis of an infected goat herd in Canada. What we found is that several, so in this case, 7 infected premises were found as a part of our trace out investigation. So when we looked at the animals that were moving on and off of this farm, and we did our investigation, what we found is that there are several other farms that were impacted. Same idea, like there was one herd here in 2016 in Ontario. And then in 2017, 2018, same idea. We had the index herd, which was in Manitoba. And then from doing our tracing investigation, we found several other herds. 2018, there are 2 herds together linked. The way we do our epidemiological investigations, there's the farm where the positive animal lived, and then we also look at the birth farm for that animal. In 2019, there were 2 positive flocks. There were sheep flocks. There have been no positive herds in 2020 or 2021 to date, but our surveillance numbers have been down significantly since COVID-19, especially in Ontario, where the largest amount of small animals either live or are slaughtered here in Canada. So I'm just going to sort of shift into control now. One of our biggest tools for finding scrapie in order for us to be able to eradicate it is our surveillance plan. And since I've taken over the file, I've sort of taken a look at the direction we need to go. And there are 2 main areas I've put in a project with our internal epidemiologists to ramp up our surveillance in these areas over the next 5 years. So if we're going to eradicate scrapie from Canada, we must find any remaining pockets of disease. And then, because our goal is eradication or stamping out, we need to completely clean up the farms. The 2 main areas that we still are missing adequate samples, not only just to be confident that we found the disease, also to show this information of adequate sampling scientifically, justifiably, information to our trading partners in the OIE, as well as the International Organization for Animal Health. We need to show that we have done adequate sampling in all populations necessary to be confident that we aren't going to export a scrapie positive animal. And so 2 main areas are to target certain geographically underrepresented areas of the country. We're working on gathering all of that data previously. So for where we've sampled, where the positive cases have been, where the abattoirs are, so that we can target getting samples from farms in all areas of Canada. And the second piece that's most relevant to everyone here on this call is, like I said, we have a huge shortage of information in goat samples. And so we are planning on increasing our mature goat surveillance. And so when I say mature goats, this is a really important distinction. In order to be able to test reliably for scrapie, we need to test animals that are 12 months and older. So these are clinically normal animals or on farm deads. And so we really need to ramp that up in the next several years. So we're working on, as I mentioned, past surveillance data, mapping it out, abattoir locations. And one of the other pieces, in which I really appreciated Erin and Marlene inviting me for this talk, is that it's really important to me to work on doing some outreach. I'm getting to know the various sectors of the goat industry in Canada, goat producers, and us finding ways to get into the abattoirs and collect more of those mature goat samples. And when you have an animal that dies on farm, we have a national surveillance program. So, at no cost to you, the CFIA is either able to, come out to your farm as logistically feasible to pick up the sample and bring it in for Scrapie testing, testing the heads, the brain and lymph node, or, you can call your local district office. You can drop off either the head or the carcass of an animal that dies, depending on what arrangement you make with them. We can expect, so along with this, we want to find the missing cases of Scrapie, because until we do that and do our tracing investigations, we won't have found all the cases in order to be able to move towards eradication. So we can all expect, although we haven't had cases last year due to COVID and haven't had any so far this year, we can expect the case numbers to go up as we want to find the remaining cases in Canada and stamp them out over time so that we can be declared Scrapie free. And so that is our goal. We want to find the remaining cases. So don't be surprised if those numbers do go up over the next few years. So I'm just going to really touch on, as far as our disease control actions on scrapie infected premises, what's really relevant to my talk today. When we have a positive herd, our current policy is to destroy infected and at-risk animals. And so in sheep, we've had the tool to help determine risk of genotyping. So we have been destroying the susceptible sheep, and the resistant sheep have not been destroyed. We have not had the same information. And, we still don't. We have some improved information on resistance in goats, which I'll share shortly. So our current policy, our official policy, is complete herd destruction. And then we do post-mortem Scrapie testing, because currently that is the only reliable way to know if an individual animal is positive or negative for Scrapie. And then, of course, when your animals are ordered destroyed, there is compensation from the government. I won't go into too much detail on this slide, but basically, in order to test, do genotype testing for Scrapie, we currently use a blood test. And so we have this chart, this very clear linkage of susceptibility for the two genotypes at 136 and 171 in sheep. In North America, we haven't had our sheep test positive for Scrapie. In Canada, we've not had an AAQR sheep, which is the very low risk level, come up positive to date. This is just a picture of practically on farm how our current policy in sheep would work. We've genotyped them, and so we've labeled there with the blue spray paint the susceptible sheep, the ones we ordered destroyed, and the ones with the orange marks, the orange paint, are the resistant sheep who would remain on premises, and we would continue our cleaning and disinfection plan, that sort of thing. So what do we know about Scrapie genotypes in goats? In recent years, there's been convincing evidence from North America and European studies that there is genetic resistance for Scrapie in goats. Based on the data, goats having a single copy of either one S146, so it's serine at position 146, or K22, that's lysine, have shown a strong degree of Scrapie resistance to natural infection with classical Scrapie. Other alleles such as Q211, they've been studied in Europe and other countries, but at this time there isn't enough evidence to use them as regulatory purposes. As always, though, there's always research ongoing. We're always paying attention and analyzing that research. Scientists are always in contact with other researchers around the world, especially with our trading partners in the US and Europe who are doing work on this as well. And, we're always paying attention as new information unfolds. So just a little bit about some of the information Dr. Mitchell at our OA Reference Lab has collected over the last several years on these alleles in Canadian goat breeds. So S146 is the most common allele for scrapie resistance found in meat breeds, such as boar and Savannah, as well as Nubian dairy goats. The K222 allele has been found less often in goats tested up to now, mainly in Toggenburg herds. And these alleles are most likely to be found in other breeds in Canada, just as we're able to genotype more goats. Internationally, here's what we've seen. And, as we know, there is some information that carries over and crosses over internationally, and some of it, a lot of it depends on our own use in our own populations. We have different variants, that sort of thing, of disease in different areas of the world. But this is what we found internationally. So allele S146 has been found in meat and dairy breeds, including boar, Nubian Alpine, Saanen, and La Mancha. The K22 allele appeared most often in dairy breeds. And so Toggenburg, La Mancha, Saanen, and Alpine. How does this apply to the goat industry? So at this time, in line with our trading partners, we've had, of course, these discussions behind the scenes in our work with them. In line with the US, we are most closely aligned with them as trading partners currently. The CFIA recommends focusing on alleles S146 and K22 while breeding for resistance in goats. And again, that is in line with the latest international information we have, as well as our domestic experience. We are happy to announce that in Canada, there are now genotyping services available for DNA testing at these two alleles, available through GoatGen or Animal Health Laboratory. If you'd like more information about that, we do have a public notice with links to their websites, so that can be made available through Marlene or Aaron if you would like that information. I can send a link to that release. And so I'm most definitely, although I am a veterinarian and not an expert in breeding programs, but I did just want to make this point while I'm here. At this point, based on the information we have on genotyping, goats producers may use genotyping as part of their selective breeding programs and for individual herd management decisions. So at this time, the genotyping results for external laboratories, they're not considered official. If we have, a positive herd, we would have to do our own testing through the CFIA during a disease response. But certainly they can be used for breeding programs and your individual herd management. The thing I wanted to mention here, and I really appreciated Aaron's point about breeding and you getting your progress over time. Just that it doesn't have to be all or nothing. I mean, certainly when you breed for scrapie resistance, it's going to also depend on your overall goals, right, for your business. And just that it doesn't have to be all or nothing. So you can look at conformation, production traits, reproduction, and scrapie resistance. And again, whatever your goals are, you can look at making progress in your herd over time. And so next steps, I just wrap up by talking about some of the work we're doing at CFIA as far as a pilot project. What we want to understand, this is under development, so I don't have a lot of details. Of course, I work in the policy and programs branch, and we need to work with our colleagues in science and primarily our operations branch who would carry out the pilot project on farm when we have an eligible herd. And so the pilot project will apply to positive goat herds. So first we need to find some positive goat herds and we'll then see if they're eligible and hopefully they can participate in a pilot project. And what we want to look at is assessing an alternate disease control pathway from our standard disease control procedures, which is total herd destruction currently, if we have positive goat herd, involving genotyping along with other tools that we have available at our disposal. So what we're going to look at is an alternative to the current whole herd destruction in positive goats and assess if that is adequate to eradicate scrapie on that farm. Because just remember, we are trying to eliminate scrapie from the country. So what we need to understand, I showed you that chart and it had very clear ranges of categories of susceptibility. We don't have that yet for goats. So we don't understand how susceptible they are depending on the genotypes, as well as like how contaminated is the environment. What is the prevalence in the herd? Do we have a highly infected herd? What impact do the genetics of the animals in the herd have as compared to how infected the herd is? So those are things we have a much clearer understanding of in sheep. We do not have that information yet in goats, and that's what we hope to accomplish. It would be nice if we could eventually have a chart like that, so we have a clear delineation of what genotypes or what animals are susceptible, have a low risk, or are highly at risk of contracting Scrapie. So why a pilot project? Based on the internationally accepted data and us having enough Canadian data now, we can look at using Scrapie resistant genotypes to sort of triage goats in our disease response, similar to the way we explored this in sheep. Other Scrapie affected countries have explored these sorts of deviations from their standard programs in recognition of new information, and it has been specifically related to alleles S146 and K222. So any derogation, which means any deviation from our standard program, which is the current total herd destruction, would adhere to these following principles. That the health of our national flock is not compromised. So we have to remember, first and foremost, our goal is to eradicate Scrapie in Canada. So we do not want to leave infected animals or infected farms behind. The welfare of goats on Scrapie infected premises, we have to make sure that it is not compromised. And again, it comes back to us being confident that the actions we take are still in line with the goal of eradication, and we don't leave behind any infected animals or farms. As producers, how can you help to eradicate Scrapie? Here are a few ways. Very, very important. In order for us to test animals, one of the things that's going to help us trace that positive animal back is we need to be able to identify them. So while a mandatory national ID isn't in place, it was unfortunately delayed due to COVID. It was supposed to be on the books for this year. I think it will be next year now. But in the meantime, there is a list of pre-approved identifiers. And the government is open to members of the goat industry doing tag trials, that sort of thing. I'm ongoing to continue to add to that list to find identification solutions that will work well for your industry. But at this point, it is voluntary, and we encourage as many producers as you can to voluntarily identify your animals. And along with that, to have as many brains and lymph nodes of mature goats, so greater than 12 months, tested that die on farm or are slaughtered. One of the challenges we have, I'll just share this as a little bit of a side note, and I'm not sure exactly where the problem lies, so I'm definitely following up on the abattoir side, is that when we get the reporting of the few goats and kids in the grand scheme of things that are slaughtered in Canada, we don't get differentiation of how many were mature versus kids. And so we can't use that data because what we're looking at is the pool of eligible animals to be Scrapie tested, and they need to be 12 months or older in non-clinical animals for us to be confident in the test results. So one thing you can do as a producer is to indicate your animal's age when the intake is there, and request that information is shared on the submission form. And like I said, from my end, I'm following up through CFIA and the abattoirs to try to have better differentiation when animals are slaughtered, whether they're mature or immature animals. I won't add anything to the great presentation that Marlene and Aaron did, but definitely, looking at reducing inbreeding and incorporating, along with your other goals, Scrapie genotyping into your selective breeding programs over time. And the other piece is to join the Scrapie flock certification program, or to help manage the risk in your herd by goats from producers on the Scrapie flock certification program. That's all I have. Hopefully you have a few minutes for questions. Feel free to either put your hand up or pose a question in the chat. I can't see everyone, so I'm going to rely on Marlene and Erin and Oshie to share any questions with me.
Yeah, thanks, Amanda. That was great overview in terms of Scrapies in Canada.
Thank you for having me.
Thank you, Amanda.
Take care.